Yes, great. So today I'm going to talk about Firebase, which is a pathogen host interactions database, and PHI-Canto, which is a new author curation tool that we've been developing. So I thought I'd introduce the Firebase team because it's very much a team effort. I thought I'd put some photos up as we're not all walking around in the building at the moment to see each other. So there's myself. I'm working on curation for Firebase and also developing a new phenotype ontology. We have James Seeger, who's a software developer who's working on the development of this new Canto tool. Martin, who's the Firebase database manager, and Kim, who's the PI for the project. So we're funded by the BBSRC. It's fortunate that we've now had 3 grants from the Bioinformatics and Biological Resource Fund for Firebase. And the title of this grant is A Fair Community Resource for Pathogens, Hosts and Their Interactions to Enhance Global Food Security and Human Health.
I think the problem may be that you are sharing your screen and not just the presentation, because we can see also part of your desktop at the bottom.
Ah, okay, let me...
So if you only share the presentation, then that will disappear.
Okay. Ah, that's better. Okay, thank you. It may be a bit tricky when I come to the live demos. We'll see how it goes when I get there. Okay, thanks.
You're welcome.
So, as I just was saying, this is a grant about FAIR data, and this might be new for some of you. So, the FAIR principles are making your data findable, accessible, interoperable, and reusable. It's a 3-year grant, which we're about halfway through now, and it was pretty well funded. That's because it's a grant that goes across 3 different sites. So we're based at Rothamsted. We've got the Firebase team and the Netminer team. Then we also have the Pubmed and the Canto team at the University of Cambridge, and then we're also linked with EBI and Ensemble Genomes in this grant. Can't click through my slides. Okay. So today I'm planning on giving an introduction to Firebase. I'm going to talk a bit about the development of the PHI-Canto tool and hopefully give you a live demo. And then going to describe some plans for a new Firebase interface and then some other future plans. So Firebase is a pathogen host interactions database. It contains data from genes to mutant phenotypes and is available at firebase.org. So it's been online now since 2005, so a pretty long time. And it contains data that's manually curated from scientific literature by 2 professional curators that we have. It's a multi-species database for fungi, protists and bacterial pathogens. And at the moment, it contains about 60% of plant pathogen data and 40% of human, animal, fish and insect pathogens. So Firebase is a knowledge base. Users can look up phenotype and molecular data online. We have over 13,000 users a year from 130 countries, and we have 2 data releases a year. So our most recent release was in November 2020, and that was called version 4.10. It contains data from 3,914 papers at the moment. We've got 7,681 genes in there, nearly 16,000 interactions, and 274 pathogens, 416 hosts, and 568 diseases. So the Firebase data is fair, and we provide phenotype annotation to a variety of other resources, including the EBI, FungiDB, Netminer, Uniprot, and Elixir. The top 15 pathogen species in Firebase are highlighted here as little images. In green, we have plant pathogens, and those include Fusarium graminearum, Agnotha oryzae, and then the rest there in the images. We also have animal pathogens outlined in red, such as Salmonella, Candida albicans, Aspergillus, fumigatus. And then we have this case, which is a Pseudomonas aeruginosa that can actually infect both plant and animal pathogens, which is quite interesting. So looking at the content of Firebase from a designing future wheat focus, we have about 12% of the entries are from wheat hosts, which is quite interesting. We have 1,670 entries for Fusarium graminearum, 78 entries for Zymos septoria, and 3 for Takel. So we recently published a Firebase update in nucleic acids research, if you want to go and find any more information about that. So what does Firebase look like? Well, if you go to the website, you can type in your gene of interest into the search bar at position 1 here. So here we have Tri5, which is a gene involved in DON mycotoxin biosynthesis and Fusarium graminearum. It's produced a couple of summary results here, which are the rows of information. Hopefully you can see my pointer. If you click on the row, you can open it up, and then we have further information in the tabs here. So at point number 2, we have information on the pathogen gene. So we have the Tri5 gene, we have the Firebase entry ID, protein ID. Then in the next tab over, it's about the pathogen species, so it's Fusarium graminearum. We have the NCBI taxonomy ID linked with this species, and we have the experimental strain, so that's PH1 in this case. Next over, we have the host information, so the host species, so we've got Triticum here, we've got the NCBI taxonomy ID and the strain Bob White. Then in the reference tab, we have the Pubmed ID and information about the year the paper was published and the authors. And then we have the phyphenotype tab. So in this case, we have the Tri5 gene, which has been deleted in Fusarium graminearum and inoculated onto wheat. And the phenotype that was observed was reduced virulence. So this is one of 9 high-level terms that are listed here that we've published previously in 2015. And this phenotype was observed on spike tissue in this particular experiment. Over here in point 4, it's possible to refine the search. So if you have a lot of entries, you can click on things such as below the host species, you could limit it to just the Arabidopsis or just the wheat entry if required. And then up at point 5, we have a Firebase blast tool, so you can blast your favourite gene against the contents of Firebase. So Firebase is part of a connected network of FAIR data resources, and we currently provide data to a variety of these resources, such as FungiDB, Ensemble Genomes, Uniprot, and the Disease Netminer. So how do we get data into Firebase? Well, at the moment we've been using a professional curation company in India called Molecular Connections, and we have 2 curators that do this work for us. So the workflow is as follows. Molecular Connections will do a keyword search in PubMed to generate a list of candidate articles. They will send them to us here at Rothamsted, and we will review this list, add in anything extra that we feel is missing, collect these PDFs together, and then send them back out to Molecular Connections. They will then curate these papers at a rate of about 10 papers a week, and then they send them back to us in monthly batches. So this curation is currently happening in Excel. We have a very large Excel spreadsheet at the moment. And for each entry for each gene, there are up to 80 columns of data that can be collected. This data is then reviewed, and we prepare it so that we can make 2 database releases a year, each of about 6 months of data. Once this is prepared in Excel and the data has been cleaned up, it gets parsed and it gets released onto our Firebase web page. So 85% of the Firebase data has now been curated by Molecular Connections, so they've really had a big input into our data collection here. They have curated 3,400 papers since they started working on this project in 2012, and that was 480 papers alone for last year. I'm now going to move on to talking about development of the PHI-Canto tool. So this is the Pathogen Host Interactions Community Annotation tool. So PHI-Canto is a multi-species author curation tool. Here's an image of the font page. You could start by just adding the Pubmed ID for your paper and then following 6 easy steps, which I'll give you a bit more information on as we go through the talk. This was developed in collaboration with the Pubmed team at the University of Cambridge. So Firebase is a fission use phenotype database that's managed by Varwood. Kim Rutherford has been working with them to develop Canto, which is their online curation tool. So community curation has been successful for the fission used community. I've taken this graph from one of their web pages. In the green bars here, these are the publications that have been curated directly by the Firebase curators since the 80s into Firebase. And then from about the 1990s, you can see that there's a few grey bars where there seems to be an increasing number of uncurated papers. So it looks as if they've had a data deluge and there's too much for them to capture. In 2009, they started a pilot curation project and developed the Canto tool. And what they started doing was actually asking the community to curate their own papers. So they would assign the papers to the authors to curate, which are the bars in red. And then once those authors had curated them, they've been recorded as blue bars here. And now since 2012, 1,716 publications have been assigned for curation, and 890 of these have now been curated by the authors. So Val is really pleased with this number, that they have now reached 52% where the fission use community curate their own papers. So for Firebase, we're now dealing with a similar data deluge. Over the years, the number of publications with pathogen host interaction related data has really increased. And we feel that we also need to have our own curation tool, and that's to keep on top of this data deluge, but also to capture the greater detail in these papers, because some of them are really becoming quite complicated and we've been unable to capture all of the detail, particularly on the host side in the past. So there were 4 key tasks that we had to undertake to adapt the Firebase Canto from the single species community annotation tool to the multi-species tool that we needed for PHI-Canto. So firstly, we needed to enable a Uniprot adapter module. Secondly, we needed to create the concept of a metagenotype. Thirdly, we needed to create FIPO, which is a pathogen host interactions phenotype ontology. And fourthly, we needed to create some additional control vocabulary lists. So I'll expand briefly on each of these points now. So the first task was enabling the Uniprot adapter module. This was to retrieve gene and species information from multiple species. This had not been required in Canto, just for the fission used on its own. For those of you that aren't familiar with Uniprot, Uniprot is a resource which catalogues proteins and provides them with unique IDs, and it's available at this website here. I've just got a screenshot of an example. So I was looking at the GT2 gene from Zymo-Septoria tritici in Uniprot, and below in the red oval we can see that there's a main Uniprot ID that is pulled back in the results. So this information can be pulled into PHI-Canto using this ID, and we can capture things that Uniprot have, such as the protein name, the gene name, and the organism species of interest. The second task was creating the concept of the metagenotype, and this is to capture the combined pathogen and host genotypes that we use in our pathogen-host interaction experiments. So we needed a way of recording what the pathogen genotype was and the host genotype, and then combining them into a metagenotype, which will represent the majority of the experiments that are done in either the lab's growth room field. Once we have this metagenotype in place, then it's possible to annotate that with a phenotype that's been observed, and that phenotype will come from the FIPO ontology. So the third task was development of this FIPO ontology. So it's the pathogen-host interaction phenotype ontology. An ontology provides precise definitions for things, such as classes or terms, and the relationships between these things within a given topical domain. So it's a hierarchy of parent-child terms which are linked by an is-a relationship. Here we have a screenshot of FIPO. So I've been using Protégé, which is a free online ontology development tool. And there are two main branches that we've split the ontology into for FIPO. We have the pathogen-host interaction phenotype, which is currently closed up at the little triangle. And then we have the single species phenotype branch, and that captures the pathogen-only and host-only phenotypes. So that branch is opened up here, so you can see some of the hierarchy. So we have decreased hyphal growth is abnormal hyphal growth is an hyphal growth phenotype, and that just kind of goes up the hierarchy. So I've been working on this ontology with Val, and some of it's closely following the ontology that they developed for fission use phenotypes. So each of these ontology terms is human readable, so the terms have a clear text definition for the curators to read and fully understand, and is also computer readable with logical definitions using design patterns developed with the UPheno team at the EBI, and this is work that James has been doing as part of the Monarch initiative. So FIPO can be used by others as it's registered with the OBO Foundry. So the OBO Foundry is where a lot of biological ontologies are registered, such as the gene ontology, and that also helps to make our data fair. The fourth main task was creation of additional controlled vocabulary lists. So these included FIDO, which is the pathogen-host interaction disease ontology, PHI-eco, the pathogen-host interaction experimental conditions ontology, and then we needed some experimental evidence codes, pathogen, species, strains, and also host species strains. And all of these controlled vocabulary lists are required for the successful curation and to ensure all of our data is fair. So the types of data that can be curated into PHI-Canto. So at the top left here, we are capturing information about the gene, the pathogen species and the host species. And that can be captured by the Uniprot ID, which pulls this information in. We can also curate a variety of different phenotypes. So we can curate pathogen phenotypes, host phenotypes, pathogen-host interaction phenotypes, and more recently gene-for-gene interaction phenotypes. And this data is created by annotating a phenotype term from FIPO to the genotype. Additional information can be curated for Go molecular function, Go biological process, Go cellular component, and also if there's a physical interaction between the two proteins or a protein modification. So these will be annotated with terms from external sources such as the gene ontology, and they are annotated to the gene or Uniprot ID rather than the genotype, which usually refers to the mutant alleles. So I'm going to run through some examples looking at curating pathogen phenotypes and pathogen-host interaction phenotypes today. So firstly, I'm going to explain the 6-step process of making a phenotype annotation. So in step 1, you enter the Pubmed ID for your publication. Then you enter your curated details, so that's your name and your email address. Then you need to add in your gene, species and strain information. The next step is to create your genotype. So that could be a single species mutant genotype or a pathogen host interaction metagenotype. And as I mentioned earlier, the metagenotype is a combination of the pathogen genotype and the host genotype. So for the pathogen genotype, that can be broken down into the strain information, any background mutations, for example, Ku70 enzyme septoria, and then the modification to the gene of interest. So that could be a whole gene deletion or an amino acid substitution. On the host side, we also capture the strain information, background mutations if relevant, and modification to the gene of interest if relevant. But in many cases, it's a completely wild type host that's been used in the experiment. Once we've created our genotypes, we can then select the observed phenotype term from FIPO to annotate to the genotype. Then we can select our experimental evidence and conditions. We can select any additional information that we want to add as an extension, such as the host tissue that was infected within the experiment. Then the data can be reviewed, you can add a comment, and then you can submit it for checking by the PHI-Canto team before it would be visible on any websites. So we chose 10 example papers covering a really wide selection of literature, and that was to seed the FIPO terms and develop the curation techniques. So these are just the list of the types of experiments in these papers. So we have a paper on receptor decoys, on RR-AVR interactions, secondary metabolite clusters, early acting virulence proteins, first host targets of pathogen effectors, fungal toxins required for virulence on plants, bacteria-human interactions, fungal-human interactions, a mutualism interaction, resistance to antifungal chemistries. So as you can see just from this list here, that FIPO has to cover a really large range of paper types. And I'm going to show you an example of a paper covering early acting virulence proteins. So the paper I've chosen is a conserved fungal glycosyltransferase facilitates pathogenesis of plants by enabling hyphal growth on solid surfaces. So we've got some photos here. You should recognise these people. Rob, Martin and Jason were the authors on this paper. And in the left-hand panel, you can see that Zymo septoria is just growing as a wild-type Zymo is growing on a plate here. And in the right-hand panel, there is a GT2 deletion mutant, and there's a clear phenotype there. So now I'm going to talk through making annotating a pathogen phenotype to a pathogen genotype. This next section is a summary of the annotation that I've snipped out of PHI-Canto, and you can see under the pathogen phenotype section we have the information for this particular annotation. We have the species information, so it's Zymo septoria, and the strain is IPA323. The gene that we're interested in is GT2. The genotype is delta GT2, so it's a deletion mutant. And this has been annotated with the FIPO term decreased hyphal growth, as observed in the image. There's a term ID that's also linked with this as part of FIPO. We've captured the evidence as a growth assay. We've recorded additional conditions like it was water medium and agar plates. And then we've also recorded that this was actually observed in figure 2E, which makes it much easier to go to the publication and see exactly which piece of information is being recorded. As you can see in this annotation, we have used several of the control vocabulary lists, including the strain information for the IPO323, the phenotypes for the FIPO term, the evidence code, and the conditions. So it was really important to develop all of these before we could use the tool successfully. So here's an image of FIPO and where this FIPO term decreased hyphal growth fits into the hierarchy. It's in the single species phenotype branch. So each of these phenotype terms has its own term ID. So in this case it's FIPO 0001212. And next to that there's a link for a Perl oboe foundry, and it has the FIPO ID, and that's this persistent identifier for this particular link, for this particular phenotype term. The term label in this case is decreased hyphal growth, and then we have the definition, which is what the curators can read. So an individual organism growth phenotype in which the growth of the hyphae is decreased. So each of the terms in FIPO has all of this information. The second example I wanted to show you was annotating a pathogen-host interaction phenotype to a metagenotype. So in the left-hand image here, we have the wild type Zomoseptoria that's now being inoculated onto wheat leaves. This can be captured in this annotation. So under pathogen genotype, we have GT2+, which indicates it's the wild type Zomoseptoria IPO323 strain, and that's on the host genotype, which is wild type Triticum, and it's a cultivar riband. That's been annotated with the FIPO term presence of pathogen-associated host lesions. The evidence has been added, and the conditions, the figure information, and then some annotation extensions in this case. So we can see that it infects leaf tissue, causes septoria leaf blotch, and that the disease is present. So that's the control annotation. Then when you inoculate the GT2 mutant onto wheat, we have a change in phenotype. So we can run through here. Under the pathogen genotype, it now shows the GT2 deletion. The host genotype is the same, and then we have a different phenotype term, so it's now absence of pathogen-associated host lesions. The evidence, conditions, and figure information are the same, but the annotation extensions have changed so that we have captured that we have a loss of pathogenicity in this case, which is one of our 9 high-level Firebase terms I mentioned earlier. And this is now compared to the control, the GT2 wild-type control metagenotype entry above. This is the location of these FIPO terms. The absence of pathogen-associated host lesions and presence of pathogen-associated host lesions are both in the hierarchy underneath the pathogen-host interaction phenotype branch. Each of those will have a term ID and a definition as well. I'm now hoping to show you a live demo on PHI-Canto of those annotations. I'll just leave this. Are you able to see
yes.
web pages? Great, thank you. So this is the demo site for PHI-Canto. At the top right hand corner we have some help documentation. So we have a nice table of contents here. You can click through. This is about getting started with your sessions and read through this. And then, as I said earlier, to start your curation session, you need to enter a Pubmed ID for your paper. So earlier on, I went to Pubmed and I searched for the paper that we're curating. You can grab the Pubmed ID from here and enter that into this section here. Press find. So that's now pulling up the paper that we were curating, so you can check that the title and the abstract and the authors are as expected, and we can continue to the next page. It's possible to reassign the curation to somebody else, but we're going to do it ourselves, so I'll continue here. This is where we can capture information about who's curating, so you enter your own name, your email address, and an ORCID ID if you would like to, but for this demo I'm just going to stick with these demo information. So for the paper that we're using, we have an example where we have one pathogen gene that's inoculated onto one wild-type host. So at this point I'm going to enter the information about the pathogen gene and then the host. So for the pathogen gene I need to get a Uniprot identifier. So we go to our Uniprot page, and I have already looked up the GT2 gene as Septoria and have got the Uniprot ID here. I'm just going to copy that in to the box. You can add, if your paper has lots of genes, you can just list them all in here in one go. So now I'm going to add information about the host organism. So we were inoculating onto wheat. So we can put that in here, and we come up with both the species name, the common name, and the NCBI taxonomy identifier. On this page we can just check that we've got everything we expect to have. So under pathogen genes, it's the correct organisms Septoria. That's the Uniprot ID that we entered. We have the gene name, and then we have the gene product. I'm going to now enter the information about the particular strain, because as we're very aware, it's very important to know which strain of each species we're looking at in the experiments, because it can make a big difference on the outcome. This is the strain list for Septoria that I mentioned. We had to create this for each of the species we have in Firebase. So now I've added IPA 323. Now in the host section we have Triticum as expected. We don't have any specific genes we're looking at, we're just looking at the whole of the wheat plant in this case, so there's no Uniprot IDs required. But we do know that the cultivar ribbon was used in this experiment, so I'll add that here. Now we come to our Pubmed Summary page. So on the left hand side we have the information about the genes and species we entered. So we have our pathogen Septoria and our GT2 gene. Next to it we have our host Triticum. On the right hand side of the page we have the publication details, just to remind us that we're in the right session here. And the first example I wanted to show you was making a pathogen genotype and annotating a phenotype. So I've gone into pathogen genotype management. We can see our pathogen here is Septoria, the gene is GT2. So to begin with I'm going to make a wild type genotype for this. So I'll click on here, select the allele type, so we have a variety of alleles that we can choose from. I'm going to choose wild type. The allele name is automatically added, so it's GT2 plus to indicate it's wild type. It's recorded the strain that we have. If we had multiple different strains in the experiment, we could toggle between those. It's a wild type allele, and therefore it's a wild type allele expression, so I'll select that option. And now we have our single locus genotype created. So this is the GT2 wild type allele with a strain. I'm going to make a second genotype now, and I'm going to use the quick deletion option here. And that's the strain we're using. And that gives us GT2 delta, which is our GT2 deletion mutant in IPA 323. So next I'm going to annotate a phenotype to this GT2 deletion mutant. So I will click on the Starter Pathogen Phenotype Annotation link. Up here it reminds us that we're annotating the GT2 delta genotype. I'm going to search here now for a pathogen phenotype term. Hyphal growth. So you can see here we have our FIPO term ID, and then we have a nice text definition just to confirm that it's exactly as we wanted it to be. Collect on that. Then the tool provides us with an option to either choose a more specific child term if we wanted to, like abolished hyphal growth, but we don't want to do that in this case. And in some cases there may not be a FIPO term that matches your requirements, so you can actually suggest a new term, but we don't need to do that either here. The next stage is choosing the experimental evidence and conditions. So we have a selection here. I'm going to choose a growth assay. The experimental conditions were that it was water, medium and agar plates that this was grown on. This is an option to add annotation extensions, but for this example I won't be doing that, so we'll skip that section. So we can see now that our annotation is building up. There's next a place where we can capture the figure, so we observed these results from figure 2E in the publication. If there's any comments or extra information we were unable to fit in, we can put this into this comment box here, but I'm not going to use that in this example. So now under the pathogen phenotype grouping, we've made our annotation. So we have our Xanroseptoria IPA323. The gene we're looking at was GT2, and it was a GT2 delta, so it's the deletion mutant, and that's been annotated with the decreased hyphal growth term. We've selected our evidence code, our experimental conditions, and we've indicated which figure we found that in. So for the second example, we're going to look at annotating the pathogen host phenotype interaction. So we need to go into metagenotype management here. So on the left hand side we have the information about the pathogen. So we have Xanroseptoria tritici, and we have the genotypes that we created earlier. So I'm going to take the GT2 wild type genotype and combine that with the host information. So on the right hand side here we have the triticum host. It's a wild type, we don't have any specified genes, but we do know that we're using cultivar ribbons in this case. So by clicking both of those buttons I can then make the metagenotype, and below here this information is now available under the pathogen part where we've got the species and strain information and the genotype, and then the host where we have the species information and the genotype, and that's combined now as a single metagenotype. I'm going to do the same with the GT2 deletion and the host, and that builds up underneath as a list. So next I'm going to annotate a pathogen host interaction phenotype to, essentially this is a control metagenotype. We have the wild type GT2 and the wild type host. Again, there's a reminder here of the exact metagenotype that we're annotating. So then in this box I'm now going to select for my phenotype term of pathogen associated host lesions. Again, we have the FIPO ID and we have a nice definition. We don't need to use any more specific terms or suggest any child terms. We'll choose our experimental evidence, and this was a macroscopic observation. With the experimental conditions, we've said that the authors looked at the 14 days post-inoculation. So that's entered here. This comes from our PHI-eco ontology that we developed. In this example, I'm going to add some of the annotation extensions. So, disease caused leaf blotch. So this comes from FIDO, which is our disease ontology list. The information is added below. I'm going to then use host tissue infected. So this was observed on leaf tissue. This term comes from the Brenda tissue ontology, which is an external ontology which is also OVO Foundry registered. We don't need any more of these specific terms here. And finally, the outcome of the interaction was that in this case disease was present. So I've now added 3 extensions, which you can see building up in this annotation. So we have our metagenotype information, our FIPO term, the evidence, the conditions and the extensions. I can add that this was also observed in figure 2E of the publication. Skip the comment section and proceed. So this is our pathogen host interaction phenotype annotation for the wild type control. I'm going to do the second annotation. I'm going to use the quick copy and edit function here. So now I would like to make an annotation to the GT2 deletion mutant inoculated into wheat, so I can toggle to that metagenotype here. I need to make an edit to the term name, so now absence of pathogen associated host lesions. Check the definition is correct. I need to make some edits to my annotation extension, so I will click the edit button. It no longer causes disease, so I will remove those. This is a mutant genotype, metagenotype, so I'm going to add compared to the control genotype, which is our wild type. The extent of infectivities, as we saw that there's loss of pathogenicity. We already have our host tissue infected leaf, and then the outcome of the interaction now is that disease is absent. I don't need to edit the evidence code or the conditions or the figure information because those are the same. So now in this section you should be able to see that that second annotation has been made. So the top one is the control metagenotype annotation. The second one is where we have the deleted GT2 gene inoculated onto wheat, and we have the differences in the annotation extensions as well. So hopefully I'm going to go back to my slide set now.
Yeah, I can see it. Yeah.
Great, thank you. So just to summarise, we just looked at a live demo of annotating a pathogen phenotype to a pathogen genotype and annotating a pathogen host interaction phenotype to a metagenotype. So PHI-Canto needs you. We have a demo site that you've just seen, which you're free to go and have a look at and have a play around with, because none of that is recorded permanently. If you have data that you would like recorded with our main tool, then please contact us and we can arrange a trial for that. Anna has already trialled PHI-Canto and she has entered her recent publication. I've been curating some of the in-house papers on Zynoseptoria, and once those are complete, I'll be asking the original authors to review those to see what they think of the annotations. And we're really looking for volunteers to curate journal club papers or a favourite paper or their own papers. People may wonder how long it takes to curate an article, and we think that if you already know the content of your publication quite well, it probably takes about an hour, but we're looking for feedback. So what are the benefits to authors of curating publications into Firebase? Well, your data will become fair, and this is something that funding bodies and journals may really insist upon in the future. You get a high visibility of your data, and you'll get persistent, unique identifiers for your data sets. The Firebase data is free to access. Some journals and other databases have got paywalls, so by entering the data into Firebase, it means more people can see your data. Using the control vocabulary terms, for example, the FIPO terms or the gene ontology terms, will make comparison across data sets more convenient and will make your data more interoperable. The curated data is submitted to other resources, as I showed you earlier in the Firebase connection diagram, so your data will be more reusable by others. We think that author curators will probably capture more detail than professional curators, and that's because they will know their own subject inside out and may have more time to spend on curating the detail. So now I just want to move on to the development of a new Firebase interface. So this is something that we've been working on with Molecular Connections, and at the moment I don't have any screenshots of the actual website. Martin and I have been making up some mock-ups and PowerPoint slides, and that's what I'm going to show you next. And the vision of this is that we would like to have a gene-centric page that captures all the information available for that given gene from multiple studies. So on the left-hand side here, we have a variety of information tabs, which I've described a bit to you earlier, capturing all the different curated data. The idea is that we would like to have a gene-centric page where all of this information is available in one long web page, and then you can click on these tabs to quickly jump to the section of interest. So where the red star is at the top, that's your initial summary page for your gene, and we will believe it will have like this grey bar at the top of the page, where we will have a new PHI gene-centric ID, followed by the Uniprot ID, the gene name, the species name, and the reference strain there. And if we jump down to the examples I showed earlier, for a pathogen phenotype there will be a section, and the FIPO term here on the cream background will be visible, so decreased hyphal growth with its FIPO term ID next to it. Underneath in blue will be the genotype information, so the GT2 deletion and Zymo IPA323 strain, and that can be opened up, and then you can find out more information such as the evidence, the conditions, and the reference. So the phenotypes will be listed, and in this publication the authors also found another phenotype for a sinusoidal or wavy filament. And this was observed in two different genotypes, so the GT2 deletion and then also in a tDNA disruption mutant of the GT2 gene. So these would be listed like this. It's possible, in this case everything is from the same paper, but it's possible that other publications will do experiments on GT2 Zynoseptoria, and those would also be included in the same page. And then moving down to the pathogen-host interaction phenotype curation, we have the FIPO term at the top, the presence of pathogen-associated host lesions. In blue we have our metagenotype that we created, the additional information available if you open up the plus box there. Here we have the annotation extension, so it gives us the extra information about the disease, the tissue type, and the interaction outcome. The same here for the second annotation for the absence of pathogen-associated host lesions. And in this case, one of the annotation extensions uses one of our high-level terms, the loss of pathogenicity. So we're trying to pull that out into a separate column to make that really clear for users. So moving forward, when we have this version 5 of Firebase, we're still hoping to submit data to the same group in Ensemble Genomes there on the left, and also submit data to the Gene Ontology and Uniprot. But what we're now hoping is that the researchers that are using Firebase would also be able to curate their own publications into PHI-Canto. PHI-Canto, as I explained earlier, uses Uniprot and Go terms, and we also feed information back, so it's beneficial both ways. So PHI-Canto will lead data into Firebase, and then PHI-po, which is our phenotype ontology, will be used by PHI-Canto, and the development of PHI-Canto will benefit from that as well, because each time we curate a new paper into PHI-Canto, we decide that we need new phenotypes, and then we can create those terms in PHI-po. So PHI-po is hosted by the Obo Foundry, as I mentioned earlier, which makes the data fair, and it means that this can be reused by other people. This is a slide that Kim wanted me to add in. We're about halfway through the project now, and we wanted to highlight the major problems that we still need to deal with. We're having some difficulty with Uniprot IDs. We've been discussing this with Dan recently, and we're still trying to resolve some of the issues. So in some cases, the entries become obsoleted and are then moved into a Unipark archive, and that means that our links are lost. Not all the genes that we're trying to curate are present in the reference genome or proteome, so that can make it a bit complicated. Sometimes Uniprot contains multiple reference genomes for some species, and at the moment PHI-Canto can only curate proteins because we need to use the Uniprot identifiers to pull the information in. We're also finding it a bit tricky for the curation of disease names. We would prefer not to do it on the individual annotation, and we would like to create that as a new curation type. In the case of high throughput experiments, when there's a paper with maybe 50 gene knockouts, it may be a bit tedious having to enter everything into PHI-Canto manually. So we were wondering about developing a bulk uploader to try and deal with that. And then we need to think about the conversion of the Firebase 4 version data into the new version 5 schema, and that's something that Molecular Connections are working on, that data transition. So the current plans, I'm soon going to start preparing data for our 4.11 Firebase release, which is due in May. That data is all being curated in the standard way using the Excel spreadsheets by Molecular Connections. I'm going to continue developing PHI-po, PHI-Canto and the controlled vocabularies by using the sample set of data that we selected, and also some of the publications within the group. We'd like to roll out some user testing within Rothamsted and externally. And then we've started writing a manuscript on the PHI-Canto tool, which we'd like to submit to Nature Communications possibly this spring. And then some longer term future plans. We'd like to devise a method to link the pathogen effectors with their first host targets in Ensemble Genomes, and that's a bit of a continuation of the past PhytoPath project. We would also like to collaborate with journals to encourage the authors to enter their data into PHI-Canto at the point of acceptance. And then we can start the path of getting people to use PHI-Canto a little bit like the Pubmed team did over the years, gradually building up the users. And onto the acknowledgements. So the Rothamsted research people, we've got the Firebase team and the Netminer team, the Pubmed Canto team at the University of Cambridge, and then the PhytoPath team at the EBI, and then our external collaborators.